So over to you, Wei Li, for
thanks, Rebecca, and thanks, Alistair, for the introduction.
Slightly different for the titles.
Okay.
So my work is to focus on the QbDD approach for design and optimization of multi-stage continuous antisolvent crystallizers. And this is a further extension for the workflow introduced by previous talk. Cameron make the crystallization workflow. So this work is an integral part of the workflow that spans from the API molecules to the final manufacture of drug substance. And my work is particularly focused on the digital crystallization component. And the aim of this study is to achieve the desired acceptable particle size for a drug substance with a limited dissolution rate by implementing a continuous antisolvent crystallization. So in this case study, we use ibuprofen, BCS class 2 drug substance as a model compound in ethanol as a solvent and water as antisolvent. And currently we face challenge to implementing the workflow due to the discrepancy may happen between the model predictions and the experimental data, and potentially due to the unaccounted crystallization phenomenon. And more the competition between different quality attributes introduced additional complexities to the multi-criteria optimization, and further increase the challenge to achieve the optimum outcomes. So given what would be expected, the QbDD approach applied in decision-making for the efficacy and the efficiency of continuous crystallization, and to employing the multi-dimensional optimization strategy to ensure the sustainable and robust process operations. And the QbDD workflow has been already introduced, detailed from Cameron's talk. And here is the outline. Here begins with the initial model parameter estimation accumulates to the final microfactory run, and aiming to ensure the consistent desired product attributes. So at the initial stage of this workflow, some of the kinetic parameters can be obtained from the literature, and also can be the others can be estimated from the sequential design of the experiment. It depends on the availability of the kinetic parameters, and also if it is a new drug, we need to perform some basic experiment to obtain the kinetics. And for this ibuprofen case study, we have a secondary and gross kinetics available, and we estimated the primary nucleation from the batch antisolvent crystallization. And the graph here shows the good agreement between the predicted and the experimental data, and the corresponding confidence interval for the reliable estimation for primary nucleation. And the solubility and metastable zone were plotted, and this allowed us to calculate the yield and the supersaturation related operating zone. And so to proceed with the workflow, the design space exploration is conducted within the model constraints and decision variables. And these constraints are designed to be optimize to optimize the operating window to avoid the issues such as the interline line transfer, which is caused by the too high solid content, and also the higher supersaturation caused the falling of agglomeration related issues. And these are all the real equipment capabilities and the operation boundaries. And finally, we have 3 key performance indicators that have been prioritized among all the quality attributes in this scenario, which including the supersaturation level, yield, and the final water content. This final water content can facilitate the future filtration and drying process. So a case study here on this at the stage 9 on decision making to the design space analysis for the single solvent antisolvent sources and multi-antisolvent sources. And we found that the multi-antisolvent sources have a satisfied operating zone in terms of the 3 key performance indicators we defined previously. And because the multi-antisolvent sources enable the reduction of the supersaturation level across each of the vessels, and effectively mitigating the issues associated with the falling or agglomeration or line blockage, which the satisfied operating zone highlighted in blue color, and with a lower supersaturation, at the same time maintaining a low water content and a higher yield. And then for another case study here, we investigated the number of the stages for the further to design and the optimization of the MSMPRs. And for similar stance, the configuration with a very number of the stages were investigated. So we found that both 3-stage and 5-stage configurations have a design space that can satisfy all the targeted quality attributes. So we conclude that the 3-stage operation is enough to meet the desired quality attributes, and also indicating an unnecessary transition to a 5-stage operation. And for further, we run a model validation. And during the model validation stage, a significant agglomeration was observed from the PVM image here, and resulting like a substantial discrepancy between the non-agglomeration models predicted data compared with experimental validated data. And also the agglomeration lead to a considerable particle size increase in this case. So consequently, we quantify the agglomeration kinetics using Momuta's agglomeration model. So according to the workflow, the primary objective of this work to address all the mechanisms that contributes to the size changing mechanisms. And if we understand the agglomeration more, so we can effectively mitigating the issues related to agglomeration. And then finally, we implemented this complete set of size mechanisms to the continuous to 2-stage validation with the presence of the agglomeration model. So on the the left-hand side is the concentration of the 2-stage, and the right-hand side is the medium size for the 2-stage. And we can see like the particle size and the concentration measured between the measured and predicted size and predicted data have a good agreement between each other. So that means the agglomeration is reliable estimated with the previous batch antisolvent crystallization. And for this stage, we continue to stage 11 with the presence of the agglomeration, and previously with just a nucleation that goes the kinetics, we use the existing seed in stock, which is not optimized. And but under the presence of agglomeration, showing that none of the configurations, regardless of the number of the stages, can satisfy the desired particle size that necessary for the dissolution rate, the required dissolution rate. So the priority of the KPIs, given the presence of the agglomeration, has shifted the focus towards the particle size reduction while maintaining the acceptable yield and the water content. So the seed size can be optimized here to the certain limit that possibly meet the KPIs. You can see the highlighted green spot here meets all the targets the KPIs for the three-stage crystallization, it can maintain a higher yield and the required particle size for dissolution rate requirements, and also a lower water content. And finally, to conclude this work, so the missing kinetics parameters, including the primary nucleation and agglomeration, were reliable estimated. And we also completed the model parameter validation for the two-stage cascade run. And for the future validation will take place in the multi-stage microfactory. And the future optimization under the presence of agglomeration will be performed to obtain the sustainable and consistent product attributes. And the design space identification was achieved based on the global sensitivity analysis for the multivariable space that can capture the impact of the CPPs and the CMAs on the targeted quality attributes. And the design space of the multi-stage and continuous MS and PR crystallizers can enable us to make a well-informed decisions to achieve the targeted API quality attributes during the initial development stage. And finally, great thanks to the Loughborough team for my supervision, and also the Strathclyde team for their support for the experimental data for validation of the modeling work. Thanks for listening. So we have a showcase and poster for the QbDD for crystallization part for more details if you want to speak to us. And also the whole QbDD workflow in showcase 6 and poster 7 for Marie Robertson's.
Thank you, Ray. Just pulling out the detail under that, sort of the individual areas that Cameron highlighted. Any questions of Ray? Just one around the way of working in terms of modelling and experiment and just getting the data that you need for the models. How can we make that process faster and think more efficient in helping to use models to try the experiments that we're doing? Or is it already optimized?
So I think it's like the workflow does work well, because you see the workflow is like if we have any missing kinetic parameters, we go back and forth for several times, and then it's a great support from the Strathclyde team, and we have like a very frequent meeting to check the modeling and how the model communicate with the experimental setup. And also the real operating constraints is need to align with the operating limits in the lab setup. Yes. So the workflow is quite standardized how we make sure they proceed to the QbDD workflow goes smoothly. Yes.
Thank you. Any questions? No. The next speaker, thanks again.
Thank you.